Welcome to the first overview on genetics, and we're going to start with meiosis. Meiosis is very similar to mitosis. We're splitting the cell this time, but this time we're going to do it twice. And this can be found in your textbook in 11.4. So the question we're trying to answer today is what events occur during each phase of meiosis? And what's really important for you is what happens, what's the result at the end of meiosis 1 and the result after meiosis 2. So the result of meiosis 1 and the result of meiosis 2. Those are going to be super important for you as we move along through the rest of this unit. And meiosis is a process in which the number of chromosomes, what's important is the number of chromosomes per cell is cut in half through the separation of our homologous chromosomes in a diploid cell. Meiosis usually involves two distinct divisions called meiosis 1 and meiosis 2, and by the end of meiosis 2, that diploid cell that we started with is going to become 4 haploid cells. So let's get into it here. Meiosis is going to be prefaced by what we call interphase 1. And during interphase 1, there's going to be a period of growth, and after that period of growth, we are going to have an S phase. And during the S phase, we have DNA replication. I reserve the right to ask you questions about DNA replication, even though that was 2 units ago. I reserve that right, so make sure that you understand DNA replication. Each replicated chromosome is going to consist of those 2 identical chromatids joined at the center. In prophase 1, this is where we're going to get some differences. In prophase 1, we have something called synapsis. And synapsis is when our 2 homologous chromosomes that have been copied are going to come together. So our chromosomes pair up. And you can see that below in the picture. We've got our chromosome from mom, our chromosome from dad, and they pair up. And this is called synapsis. When they pair up, that pair is called a tetrad. So the pair is called a tetrad. And that tetrad is going to contain all 4 chromatids. As homologous chromosomes pair up and form tetrads, they undergo a process called crossing over. So we also have crossing over. So during crossing over, we've got our tetrad that pairs up, and then the arms of those chromosomes are going to crisscross. All right, so we've got the chromatids of the homologous chromosomes cross over one another. The arms cross, and then they will actually exchange DNA. They will exchange a portion of DNA. And what that does, and it's very important, what that does is it gives us genetic diversity. And it's going to allow cells to have different combinations of DNA at the end of our division. Next, we have metaphase 1 and anaphase 1. And as prophase 1 is ending, we're going to have spindle fibers form. Spindles form. And we can see those in our picture. We've got our green spindle fibers. And they're going to attach to each tetrad. So they're going to attach to each tetrad. During metaphase 1 of meiosis, paired homologous chromosomes are going to line up. So our pairs line up. And then during anaphase 1, we are going to have our spindle fibers pull apart those homologous chromosomes. So during anaphase, we're going to pull apart homologous chromosomes. And they're going to go towards opposite poles of the cell. When anaphase 1 is complete, the separated chromosomes cluster at opposite ends. So here's anaphase 1. We've got our chromosomes being pulled towards, our homologous chromosomes being pulled towards opposite poles. So in other words, we are pulling our tetrads apart. In telophase 1 and in cytokinesis, we're going to reform a nuclear membrane. And that's going to go around each cluster of chromosomes. And cytokinesis follows telophase 1, forming 2 new cells. 2 new cells. Now, these cells are a little bit different. Our results are 2 cells, so we're going to have 2 cells called daughter cells. But these daughter cells are different than the daughter cells of mitosis. Each of these has 4 chromatids. What we see here, the cells that we see here have 4 chromatids, as it would in mitosis. However, they do not have homologous pairs. So there are no homologous pairs. We either have, for each chromosome, it's either dad's DNA or mom's DNA. We don't have one chromosome from each. And what that does is it allows for more genetic diversity. So the 2 cells being produced in meiosis 1 have different sets of chromosomes, different sets of what we call alleles. And alleles are types of genes that are different from one another. And from the diploid cell that entered meiosis 1. So this creates this different, these different amounts of alleles, these different chromosome sets. Gives us more genetic diversity. Alright, so we've gone through 1 round of division, and so we're going to start with a second round of division. In meiosis 2, 2 cells are going to be produced, the 2 cells that were produced by meiosis 1 now enter that second division. Unlike the first division, neither of those cells are going to go through a round of replication. We don't really go through interphase, so we don't have an interphase. But we are going to have each cell is going to go through a second division. So we're going to go through prophase 2. As the cell enters prophase 2, their chromosomes, each consisting of 2 chromatids, become visible. So chromosomes become visible. Visible. There we go. So we're going to become visible, and the chromosomes are not going to pair up into tetrads because we don't have homologous chromosomes this time. So no tetrads. No tetrads. So there's going to be no crossing over, no synapses. During metaphase 2, our chromosomes line up. So you can see our chromosomes lining up in both of our cells. In anaphase 2, the chromatids are going to separate. So our chromatids are separating. And then in telophase 2 in cytokinesis, and when we have an example shown here, we're going to have 4 daughter cells. Four daughter cells which are produced and receive one set of chromatids. Each daughter cell has one set of chromatids. And basically, this is known as being one N. And one N is also called being haploid. And that basically means that we don't have homologous pairs. We don't have one set from mom, one set from dad. We just have one set of chromatids. Our normal cells, our body cells, are going to be something called diploid, or 2N. And that's when we have 2 sets of chromatids, one from each parent, in all of our cells. But right here we have a haploid, 4 haploid cells now. In males, these are going to be called gametes. So here's that sentence again. These 4 daughter cells are now going to contain the haploid number of chromosomes. So in males, our haploid cells are going to be called gametes. In males, these are going to be sperm. And we're going to talk in class about spermatogenesis. And in plants, these would be like the pollen grains. In females, we're going to create the gametes' eggs. And this is called oogenesis, which we will also talk about in class. When we have 2 haploid cells fused together, we have what's called fertilization. So the fusion of 2 gametes. And this creates a new combination of alleles. Of DNA, of genes, alleles, which creates genetic diversity. So we have more genetic diversity, and the zygote is going to undergo now regular cell division by mitosis and eventually form a new organism. All right, that is it for today. I'll tell you what, the first 5 people into my room that put their name on the board will get a little treat from me, maybe a little piece of candy or something from me. So when you come into class, put your name on the board. Don't say anything. Just put your name on the board, and I'll get you something for going all the way through the video.